Want to ace your test in just a few minutes? Watch this video. What is metabolism? Metabolism is the set of chemical reactions in living organisms to maintain life. It helps organisms grow, reproduce, and respond to their environment. Who introduced the concept of metabolism? Ibn Nafis introduced the concept of metabolism. Difference between catabolism and anabolism? Catabolism breaks down large molecules releasing energy. Example, respiration. Anabolism builds large molecules using energy. Example, photosynthesis. What are enzymes? Enzymes are proteins that speed up biochemical reactions without being changed. Difference between substrate and product? Substrate, the molecule an enzyme acts on. Product, the molecule formed after the reaction. What is activation energy? Activation energy is the minimum energy needed to start a reaction. How do enzymes lower activation energy? Enzymes lower activation energy by changing the substrate shape, aligning it correctly, and reducing energy needs. Difference between intracellular and extracellular enzymes? Intracellular enzymes work inside cells. Example, glycolysis enzymes. Extracellular enzymes work outside cells. Example, pepsin in the stomach. How do enzymes lower activation energy? Diagram. Diagram shows energy reduction with and without enzymes. Who first used the term enzyme? Wilhelm Kuhn in 1878. Two characteristics of enzymes. Enzymes are proteins. They are specific for each reaction and substrate. What is an active site? The active site is the part of the enzyme where the substrate binds and the reaction occurs. What are cofactors? Cofactors are non-protein molecules or ions needed for enzyme activity. They can be inorganic, metal ions, or organic, heme. Difference between cofactors and coenzymes? Cofactors, non-protein components, can be inorganic or organic. Coenzymes, organic cofactors loosely attached to enzymes, e.g., vitamins. Difference between prosthetic groups and coenzymes? Prosthetic groups, tightly bound organic cofactors. Example, heme, coenzymes. Loosely attached organic cofactors, e.g., riboflavin. Name vitamins that act as coenzymes. Riboflavin, thiamine, folic acid. Two uses of enzymes in the food industry: convert starch into sugars for bread and buns. Help in fermentation to produce alcohol. Factors affecting enzyme activity: temperature, substrate concentration, and pH. What is optimum temperature? The temperature at which an enzyme works fastest. For humans, it's 37 degrees Celsius. What is enzyme denaturation? Denaturation is the loss of enzyme structure at high temperatures, stopping its activity. Effect of 37 degrees Celsius on bird enzymes. The enzyme activity slows down because birds have a higher body temperature. What happens if substrate increases but enzyme concentration stays constant? The reaction rate stops increasing after all enzyme active sites are full, saturation. Effect of substrate concentration on enzyme activity? At high substrate levels, the reaction rate plateaus when enzymes are saturated. What is optimum pH? The pH at which an enzyme works best. Changes in pH can stop enzyme activity. Explain the lock and key model. The enzyme and substrate have specific shapes that fit together like a lock and key. What is the induced fit model? The enzyme changes shape to fit the substrate, making it more flexible than the lock and key model. How is enzyme specificity determined? Enzyme specificity depends on the shape of the active site matching the substrate. For the full video and more short questions to complete your test preparation in just a few minutes, visit my channel and don't forget to subscribe.